Welcome, everybody. My name is Tommy Source. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today on our Purdue Lecture Hall Series, I have the pleasure of welcoming Gianna Nossa, who is exploring the mysteries of the brain with magnetic resonance microscopy. And she's coming to us from the Ulrike Didak Lab in the School of Health Sciences right here at Purdue University. Welcome, Gianna. Thank you so much for coming and telling us your story, telling us what you're doing. I'm looking forward to hearing about your research, and I know our viewers are too.
Yeah, thank you so much for having me, and I'm really excited to share a few of my projects with you guys, and a little bit about myself, of course.
Of course, of course. I think everybody wants to know a little bit of both. You
Yeah.
know, what led you to doing the work that you're doing today? And tell us, what role are you playing in doing all this work? So how about you take us from as far back as you want to take us? I'm going to shut my video camera off and I'm going to let you take the spotlight. And if you want to share your screen with us so that you can share your slides, that'd be great. And we can get going.
All right.
Perfect.
All right.
Thank
Everyone, you can see that, right?
you. Yep.
All right. Perfect. Okay, so I'll kind of explain to you a little bit about my research that kind of dives into magnetic resonance spectroscopy. So I'll provide you guys a little bit of a background on that before I jump into my projects. And then, well, before any of that, I'll tell you a little bit about myself. So I'll explain a little bit about my road to graduate school. So I actually did my undergrad studies here at Purdue. I got my bachelor's in neurobiology and physiology, and I did a minor in Spanish. During my time here at Purdue, I was involved in a couple different research experiences. My first was in looking into neuroinflammation and epilepsy. And during that time, I was doing a lot of typical wet lab work, looking into molecular changes, structural changes that occur in the brain and the hippocampus in epileptic tissue in rodents. And then I also looked into behavioral changes. So that kind of like dove into seizure monitoring and stuff like that. And then my second experience was actually a very non-traditional experience. And it kind of started out as a class, a typical, well, not typical, an atypical actually, research lab. And it was very unique in that it was open to students across multiple disciplinaries or disciplines. And it really tasked us to like join together, draw from our individual backgrounds to complete projects. And we were really only given biological and physiological backgrounds based on each individual project. So we really drew from our own experiences and our own knowledge in terms of how to complete each task. So that was really cool, and it was a very unique experience compared to a typical research lab where you're just straight up given a protocol and you just follow instructions. So that was really cool. And then at the end of the semester, we were asked if anyone wanted to present this work, because in reality, the class itself was a research experiment, and we were basically the guinea pigs. It was an experiment to kind of highlight a different way of teaching biology to students, making it more interdisciplinary, highlighting the importance of teamwork. So I actually volunteered for that, and I got to present a poster at the experimental biology in 2017, and that was my first conference. So after I graduated in 2017, I started working at the Beckman Institute at City of Hope, where I was a research associate, and I worked in a study or in a research lab looking into macrophage-mediated immunosurveillance in cancer and its potential therapies. So this research experience for me was a little bit different. So I was looking into more cell culture, and I was harvesting macrophages and culturing cancer cell strains. And we mainly worked with breast cancer, leukemia, and ovarian cancers. And then I was also tasked with a lot of the animal studies because they found out that I have very steady hands. So I was doing a lot of cancer cell engraftments. I was monitoring tumor growth and administering treatments. And then later on, I stepped up to more of a managerial duty, and I started creating protocols and lab manuals. I was in charge of safety training and protocol modifications and just stuff like that. So I was there for 2 years, and then afterwards, I actually came back to Purdue for grad school, and I joined the interdisciplinary life science program, or the Pulse program, um, with a focus in neuroscience. So I kind of wanted to step away from cancer research and dive back into neuroscience. For me, it was a little bit more interesting, and cancer was just very difficult for me to work with. um, so I came here, joined the Pulse program, and this program does rotations. So I did rotations, and I did a very good job of having very diverse experiences, and I learned a lot from all of them, and I enjoyed all my time in all of my rotations. But one of my rotations was with Dr. Joseph Spoli, and he kind of introduced me to MRI and what we can do with MRI in a research setting, and how MRI isn't just used in clinical settings. So I really enjoyed that. I really felt drawn to it. And so I joined his lab. And then for administrative reasons, I needed a co-advisor. So I actually got recommended to reach out to Ulrika Didak over in health sciences, and we really just hit it off. I actually work primarily with her now, but I'm still working within MRI and kind of focusing on the spectroscopy side of MRI, which not a lot of people know about. So I'm here to tell you a little bit more about that. So now I'll kind of jump into like the kind of projects that I'm working on. So I'm using spectroscopy to kind of get an idea of the chemical changes that occur in different neurological disorders. So I will be telling or sharing a small piece of one of my studies with you guys, and it'll be kind of looking into what spectroscopy of the cerebellum in welders exposed to manganese can tell us about changes in mood. So manganese neurotoxicity can occur from overexposure to manganese. And overexposure can occur, or usually occurs in occupational settings such as steel production, smelting, and welding. So manganese accumulates in the brain, and this can cause motor impairments, cognitive impairments, and changes in mood. And we can actually visualize this through imaging. We can see its effects in the chemical composition of the brain through spectroscopy. And then we can also see or manage or monitor exposure levels through toenails. So when welders inhale welding fumes, they're not just getting manganese, they're getting a whole slew of metals as well. It actually primarily, or the biggest concentration is of iron, but as you can see, manganese is pretty close behind. So when they inhale these metals, or the fumes, the metals tend to accumulate in the striatum and the globus pallidus. And you can see here in these images, these are these bright triangles here. So these bright areas are indication of an accumulation of manganese, and these dark areas are accumulation of iron. So all this accumulation causes mitochondrial dysfunction, and in turn leads to reactive oxygen species. And then it could also cause DNA fragmentation. So in clinical settings, this kind of, well, in extreme cases, will look like Parkinsonian-like symptoms. So patients will have tremors, posture rigidity, cognitive decline, very similar symptoms to Parkinson's disease. And what kind of happens is that there's an interruption of the GABAergic system, and this causes the disturbances in motor and cognitive function. So GABA, or gamma-aminobutyric acid, is the primary inhibitory neurotransmitter in the brain. It is involved in motor, mood, and cognitive pathways. And for the overall study, we are looking into 3 different brain regions: the cerebellum, which is responsible for coordination, movement-related motor skills, and mood; the thalamus, which is responsible for the relay of sensory signals, and this includes motor signals; and then the anterior cingulate cortex, or the ACC, which is responsible for complex cognitive functions, so empathy, impulse control, emotion, and decision making. So for this study, I will be focusing on the cerebellum. And glutathione is the primary antioxidant of the brain and is our marker for oxidative stress. So oxidative stress is a signaling response found to be increased in the presence of metal accumulation. And it kind of has a reinforcing relationship with neuroinflammation. And these are both mechanisms that have been found in Alzheimer's and Parkinson's disease. So glutathione is a cyclic pathway. So it interacts with reactive oxygen species to become oxidized. And in order to become reduced again, it requires a proton from NADPH. So when we talk about glutathione in this study, we're talking about glutathione in its reduced state, not its oxidative state. So like I said, I will be focusing on the cerebellum. So I'm showing a lot of images and a lot of images with these volumes or these squares placed into them. So I will kind of go into a little bit more about the MRI side of things really quick. So the body is primarily composed of water, and it's about 60% of your body is water, making the hydrogen atom the most abundant atom in the body. So hydrogen atoms behave like little magnets spinning in random directions. And then in the presence of a magnetic field, they will align with the magnetic field. But this isn't enough to create an image or any signal. So in order to do that, we actually present a radio frequency pulse when it's applied, it will basically flip the spins of the hydrogen atoms so that they're strained against the pull of the magnetic field. When that radio frequency pulse is turned off, the protons are slowly returning back to align with the magnetic field. And this slow release is, it releases energy, and that's the energy that is detected in MRI. And so that creates a signal. And from that signal, we can get images like this, but we can also get region-specific chemical composition. So in order to do this, we'll get an image and then we'll place a volume in our brain region of interest. So like I said, I'm interested in the cerebellum, so I put my volume here in the cerebellum. And from this, we'll place our pulse. We'll get a signal similar to an image. Our signal will go through some cool calculations, and we'll get it will transform into the frequency domain. So this right here is a large water peak. Like I said, our body is primarily made up of water. But this signal actually also has other information. It's just so much smaller compared to our water peak. So in order to actually see them, we need to suppress the water. And when we do that, we can see our spectrum much more clearly here. So this is what a typical brain spectrum looks like. Doesn't matter where you place your voxel. This is what a healthy brain will look like. So here we have a large peak for N-acetylacetate, or this is our marker for healthy neurons. We have a creatine marker. So this is our marker for energy. We have choline, which is a membrane turnover marker. We have inositol here, which is our glial cell marker. And then spread amongst these other peaks, we have glutamate and glutamine, so excitatory neurotransmitters and its precursor, as well as many other metabolites of smaller concentrations. So amongst those smaller peaks, we do have GABA and glutathione signals. And our GABA signals will resonate here in these three frequencies, sorry. And then our glutathione resonates here. So in order to calculate these, or in order to measure these metabolites of smaller concentrations more accurately, we need to edit for them. So when we edit for them, we are able to get a spectrum specifically for GABA. And here we can see a really nice GABA peak, as well as a spectrum specifically for glutathione. And we can see here our nice glutathione peak. So back to the welder study. We invite our welders here to the Purdue MRI facility, where they will undergo a brain scan, as well as a whole battery of tests. So they'll go through a psychological tests. We'll get their work history. They'll do a motor test. We'll look at their mood. They'll go through their brain scan. And then in a different appointment, we'll get their air sampling. So we'll measure the air that they're breathing in. So with the brain scan, like I said, I'm particularly interested in spectroscopy. And for the study that I will share with you guys, I will also be interested in the mood component of the study. So here in the cerebellum, we are seeing a significant decrease in GABA in our welders compared to our controls. But so far, we're not really seeing any group differences with glutathione. So one of the mood tests that the welders will undergo is the brief symptom inventory survey, and it's a questionnaire that will give us a score across multiple mood categories, and this includes the global severity index, which is our single most sensitive indicator that a person is experiencing mood symptoms. We will also get a score for obsessive compulsive behavior, for depression, for anxiety, hostility, paranoia, psychoticism, all of these. And so interestingly, we do actually see an increase in these raw scores across all categories in our controls instead of our welders, which is not something that we expected. And we do actually see significant difference in anxiety and phobic anxiety. And human research is very tricky. There can be so many components to why we see this. A couple that we suspect is, one, a lot of this data was actually collected around when COVID hit. So that does bring a lot of stress, a lot of anxieties into your lives. People were afraid of losing their jobs. They were afraid of getting sick. They were afraid of losing their loved ones. So we could understand why we would see this. Another thing is that mental health is still extremely stigmatized, especially in men. And in welding, which is a male-dominated career, we expect that maybe not many of our welders were answering the surveys as honestly. So that could also be a reason why we see lower scores in our welders compared to our controls. But as we continue to collect data, we're hoping that this will kind of balance out, and maybe we'll see a shift in these scores. So next we wanted to see if these scores were actually related to the metabolite differences. So we looked into first into glutathione. So we can see here that as glutathione is decreasing, so glutathione being our antioxidant, so as glutathione is decreasing, oxidative stress is increasing. So when we see this decrease in glutathione, we are also seeing an increase in symptoms of anxiety, as well as symptoms of obsessive compulsive behavior, phobic anxiety, and just an overall increase in the global severity score. So this could be an indicator that oxidative stress is playing a role in these mood changes. So we are also interested in seeing if neuroinflammation plays a role. And for neuroinflammation, we use the markers myonocytal, so our glial cell marker, choline, a membrane turnover marker, and total creatine, our energy marker, which all exist in higher concentrations in glial cells. So when glial cells are activated, they release pro-inflammatory signals in the brain. And so we're so when these metabolites are increased, we can assume that there's an increase in neuroinflammation. So here we can see that an increase in all of the in these three metabolites. So here we have choline, for example. We're also seeing an increase in anxiety, in depression, in phobic anxiety, psychoticism, and just overall increases in the global severity index. So this kind of tells us that we are seeing neuroinflammation, and it could be playing a role in these changes in mood. And this is not something that we are seeing in our control group either. But like I said, we do see a significant difference in GABA. However, in the cerebellum, GABA is only significantly correlated with symptoms of depression here. So as GABA is decreasing, we're seeing an increase in depression. So kind of what this means. So like I said, glutathione is negatively correlated with multiple mood categories, and this could indicate an increased oxidative stress may be associated with symptoms of anxiety and phobic anxiety, obsessive compulsive behavior. And then our markers for neuroinflammation that reflect glial cell activation. Like I said, they release neuroinflammatory mediators. And so positive correlations with these mood scores may indicate that the changes in moods could be associated with neuroinflammation. So we can conclude that mood changes may be associated with oxidative stress and neuroinflammation. And this can be reflected through metabolite changes in the cerebellum and in manganese exposed welders. So special thank you to my lab, the DIDAC lab, and my lab mates, as well as the Rispoli lab, the Purdue MRI facility, and then our collaborators, because a project this big cannot be done by a single lab. So thank you to the Park Lab, the Lou Lab, and the Foti Lab, and as well as our funding sources. Thank you.
Awesome. Awesome. That's so interesting. I love the way that you really break it down and you can let us inspect inside all the mechanics that it takes to do this type of modeling, and it's so fascinating. Tell us a little bit. What was your inspiration to get into science? What do you recall? What was the first things?
Yeah. Oh gosh. I have always been interested in science for as long as I can remember. I know that when I was 6, I wanted to be an astronaut, which, at the time you don't really consider that as a scientific field, but it is. And then as I grew up a little bit, it kind of shifted to like wanting to be a doctor. So I wanted to be a pediatrician when I was younger. And then once I got to high school, it kind of shifted a little bit. My brother, I have a younger brother, he's 2 years younger than me. He started having seizures, and um, none of the doctors or neurologists really could figure out what was wrong with him. He did multiple studies, overnight studies, sleep studies, and it was really inconclusive. So that's when it kind of shifted. Like I still wanted to go into medicine, but I wanted to go more into like neurology, and I didn't really want a situation like that to happen to anyone else. And then when I got to undergrad and I read about Dr. Brewster's research in inflammation and epilepsy, I was like, I have to do this. So I think once I joined that lab and kind of got the lab experience is when I kind of shifted from like, okay, maybe I should actually be doing research instead of going into medical school. So that was kind of my introduction to research, and really that's how I got here.
Oh, it's so awesome. So awesome because what you're really saying to me is that Dr. Brewster had a big impact on you picking research as a career option, or at least as a career option for now, right? And
Yeah.
continuing to develop your graduate studies. Tell us a little bit about her. Tell us a little bit about what kind of a mentor was she?
Oh gosh. She was honestly, she was the best. She was super sweet. She was always available to she was always, yeah, to all of her students. And she was pretty hands-on with me. And she gave me all the experiences I could have wanted as like my first research um study, I guess. Um, and even like I kept in touch with her, and she was still here when I started graduate school. So when I was starting to look into my prelim committee and my and all of that, I actually chose her as my chair. And so she really got to see me come and like start grad school again, and
So,
really saw my journey continue here.
that is so cool. That is awesome. And it's so good to hear you speaking like that because, I think a lot of people that do watch this program know that, yes, it does take my own courage to go and try the new things, or at least stick my neck out to, right? Ask
Yeah.
what could I do in your lab, or maybe how could I see myself pursuing an experience in this particular lab? What was it? Do you remember like saying like, I'm just gonna go do it, I'm gonna go call her, or I'm gonna go write, I'm gonna go, how was that?
I actually struggled finding a research lab in undergrad. So my department had somebody that would, like a coordinator that would kind of pair you with a lab. Like you would basically send them a list
Yeah.
and be like, and she would be like, okay, well, this person has like an opening in their lab and you can meet with them. And, so there was supposed to be a process like that. And I had reached out to her multiple times without any response. And at that point, I was done waiting. So I just reached out to Dr. Brewster directly. I got the tour of her lab and I explained my whole backstory and why I was so interested in her research specifically. And she was very welcoming. And yeah, I just started as soon as I could.
That's awesome. That's awesome. So, what recommendations would you have to somebody who's thinking about doing research in this area? I mean, would you recommend them going and contacting the prof directly?
Yes, I 100% I would recommend reaching out directly. Also, you want to show interest. Like it doesn't like I know I didn't really have too much knowledge in that field other than like whatever personal experience I had. But like showing interest, if you can read a couple of their papers, that is great as well. But like really you want to show that you're interested and that you're dedicated and that you're willing to learn new things always. So
Yeah.
um,
Yeah.
yeah.
And the other piece that I can learn from your story is that you might have been inspired to pursue this because of a personal experience that you had, and that might have been the motivation to get you going, right? And maybe even the motive. There's a reason why motive is in motivation, but the idea that you would have to be able to do a little self-inspection and see what inspires you, what gets you up and moving, and what gets you motivated to do sometimes the difficult and boring work. Has it all been exciting? And maybe what had, if there are people that are out there that maybe haven't gone through that personal experience, how do you get them motivated? How do they get that motivation?
Oh, well, I think it's a lot of trial and error. So definitely don't be afraid to try new things. My primary interest was in neuroscience, and so I was really lucky to have Dr. Brewster's lab as my first experience. But when I took my gap years, I did just switch gears completely and went into cancer research. And it was an amazing experience, but it was not for me. So it was very difficult to like stay in the field, and like working there just became difficult, you know? So like I learned that way, I learned the hard way that cancer research was not for me. And that's okay. That's okay if you don't enjoy a particular field. Um, so it's, yeah, I think definitely don't be afraid to try new things. Not everything will work out, but you will eventually find something that will.
I really agree with you 100%. You know, it's many times you're going to go try something and you realize, well, maybe I don't like it so much. It's
Yeah.
like, yeah, I don't want to simplify it with like ice cream flavors, but some people like chocolate, some people like raspberry or vanilla or whatever, you know, they like them all.
Yeah.
And I think it's kind of like that. It's, you don't really need to be stuck or feel like you're stuck in a place, right, Gianna?
Yeah. And there's so much going on out there. And like, even for me, like I had no idea that MRI research wasn't even an option for me. Like I wanted to do neuroscience, but like I didn't know that that was something, like I thought I was going to be doing cell culture and
Yes.
molecular biology for like the rest of my life, which there's nothing wrong with that. But I wanted to try something new, and I ended up enjoying it so much that that's the path that I chose.
That's awesome. And it's so awesome to hear it because, again, like until you put yourself out there or until you start to explore, because you might feel like a little bit uncomfortable in your situation, or maybe you're just curious about what else is out there. This field of MRI research, it's not just like, hey, how can we like use the MRI a little bit better in the clinic to take a better picture. You guys are actually like, you're really improving and innovating what we can do with the MRI, which is so amazing. It's not just a pretty picture, it's high content information, and the amount of data that you have to deal with is incredible. Tell us a little bit like how much did it take to do your thesis? Because for me, it was probably gigs, you know, maybe 10 gigs, maybe 20. But like how much how much are you using?
Um, oh gosh, I think so. We have multiple multiple like places where we store our data. We fill up a terabyte of storage very quickly.
Like a day, a terabyte a
Um,
day?
Maybe like a few months.
Right.
Yeah.
Well, this is what's been happening, right? And you are in a very different field than I was. I was more on the molecular side, and it was at a time where sequencing wasn't as open as it is now and as affordable. But still, like PhDs have gone from like collecting kilobytes to megabytes to gigabytes, terabytes, and on and on. And it's just like the amount of data is incredible. And so,
Yeah.
why is it so rich, this data, Jenna? What, like, explain a little bit about, like, what do we do with this amount of data? Why is it so rich?
Uh, well, so for spectroscopy, I don't know if it'd be the same in its other fields, but for spectroscopy, our subjects are in the scanner for an hour, which is really long. Like you would never do that in a clinical setting.
Yeah.
But we and I'm not the only one using MRI data in our lab. So the spectroscopy is my portion of it, but we have other imaging sequences as well, and my lab mates are working on those. So half of the data is my work, and then half of it is my lab mates. So it there's different, at least for spectroscopy, there's different I guess types of data files that you can save. And so when we want like the raw data, that takes up a lot of space. And we can, I don't want to say manipulate it, but we can process it differently. Um, and versus like a smaller file, which is already pre-processed through the scanner, is not like there's not much you can do with that. So for me, it's a little more complicated.
Totally. And so do you have to be savvy about databases, about computational techniques and computational approaches then to handle that data?
Um, yeah, I mean, to a certain degree, I learned a lot of it when I joined the lab. So, I mean, it's not like you're required to know it when you come. Um, but yeah, that is something that you pick up. You learn how to manage all your data. You learn how to process it the right way, and um, yeah.
And are there particular courses you would recommend if somebody were interested in doing a piece of research like you're doing? What would be courses that you would recommend or things that they could do to get prepared to be successful in that lab?
Well, my two lovely professors actually teach my two favorite classes. So Dr. Spoli actually teaches the MRI theory, and that kind of goes into a little bit more of like the physics behind MRI. And it's not just spectroscopy specific. You learn about just MRI in general and how you acquire images and how you can process it. And um, yeah, it just goes into a little bit more of the theory behind it, which is really nice and something that I really had no background in. It was super helpful. And then Dr. Didak teaches the spectroscopy course. So I basically learned everything that I'm doing right now from her class, and obviously a lot more hands-on projects as well.
Yeah.
But yeah, she goes more into spectroscopy and different methods that you can use that are available. A lot of the processing and post-processing, um, the application. Yeah.
And so where do you see yourself in 5 years' time? Do you see continuing in this area or like what's next for you?
That is a good question. 5 years from now, I have no idea. I would like to stay within the field of MRI.
Yeah.
Staying within spectroscopy would be nice, but I'm also open to learning other MRI modalities that are incredibly helpful in research as well.
Yeah.
In terms of like industry versus academia, I have no idea. I would like to pursue a postdoc to keep kind of a foot in the door for academia in case I do go into industry. I just want to keep all options available to me.
Totally, totally. That's great. That's really good. And I guess if you were going back to the time when you would be applying to university, so we're going back to high school now, and you were to think, oh gosh, I don't know what to major in. I don't know if I want to do research or not. What would you recommend to people that are in that situation?
That one's tricky because I had like my path kind of set out. I deviated from it obviously, but like I knew that I wanted to like stay. I knew that biology was an interest to me, so I went into biology, and um I'm lucky that the biology program here at Purdue, doesn't matter what branch of biology, you could study ecology, but all branches of ecology here or biology here will prepare you for the MCAT.
Yeah,
So I kind of knew when I started that like, okay, I was going to do this. This was my path. And granted, I obviously deviated from it. I did not go through with that. But I honestly like just follow what interests you. Like I know I have friends that like did their undergrad in one thing and then completely switched gears for grad school. And you can always find something that will benefit you, like whatever your experience is, like
Yeah,
it will prepare you.
totally, totally. So there is no wrong choice, right?
No.
You can just pick whatever you want, and it will benefit you. Because at the end, we all need to learn a little bit of everything in a
Yeah.
way, right?
Yeah.
I think it's fair to say that yes, it's good to be a specialist, but it's okay not to find that specialty until when you find it and what
Yeah.
you're passionate about.
Yeah.
That
And being able to adapt like that as well is such a useful skill.
and how do you do it? What like what's the best way to adapt? Because I
Oh
can see that
gosh.
you have been adapting and you've been really listening to yourself. How do you do
Honestly, I have no idea how I did it.
it?
Like, uh, I definitely going into the MRI field, like I said, I had no previous background in anything related to it, so I really dedicated myself to getting up to speed, and it definitely took a lot of hard work. I
Lots of
felt like I was,
reading.
yeah, lots of reading. I definitely felt like I was behind and like compared to my peers, but I caught up. And it like, yeah, I, anyone, I think anyone, if they're dedicated enough, anyone can do it.
Yeah, absolutely. Well, Jenna, thank you so much for taking time to talk to us and be our Purdue Lecture Hall Series spotlight member today, and wishing you all the best as you continue in your PhD and you continue with you doing your great research. And thank you again for being on the program.
Thank you. Thank you so much for having me.
Absolutely. And have a great afternoon.
Thanks, you too.
Thanks.